Good morning. Welcome to the first of the three symposia about the future of brain research. I apologize for the delay in opening the symposia. Simply, it's part of the Board of Governors meeting at the university. I was tied in to the board meeting, which was running late. So it's a great pleasure to open the symposia with Edmond, the outgoing president of the university. Understanding the human brain is the most challenging, the most significant, most fascinating scientific problem facing scientists, humanities today. It is important problem, important research subject, first because it's there. Forget about potential applications, simply understanding ourselves, understanding the mechanism of how we feel, how we see, how we interpret the world, how we react to stimulus. This is something that it's almost part of us being human is understanding how that happens. But also, it has tremendous potential useful applications. Of course, medical applications. The diseases that has to do with the operation of the human brain, with the nervous system, are some of the most debilitating diseases existing. If you talk about Alzheimer's, Parkinson, multiple sclerosis, etc., etc. This is something that creates a lot of suffering, terrible deterioration of the existence of people as a human being, and any attempt to try and deal with this issue is something which is of enormous importance. There is other potential benefits from the study of brain research. Understanding the human brain could be a model for building systems that could imitate the operation of the human brain, and that could generate very significant technological improvements. Now, this symposium has been part of the inauguration of the Edmond and Safra Center for Brain Sciences at the Hebrew University. So in one word, let me say why the Hebrew University. And I'll go back to the problem of understanding the human brain. Understanding the human brain, because it's such a complicated system, I don't know whether or not, whether it's, you could say it's the most complicated natural existing system, requires a multi-level approach, a level in which you have a description on one level, the level of, let's say, behaving organism, the level of the system of a network system of the neurons, it could be a level of the molecular level. It requires by its character, it requires a multidisciplinary approach. We will not succeed in making progress in understanding the human brain without combining the efforts of people from many different approaches and many different disciplines. I think one of the advantages of the Hebrew University is the ability to think out of the box, to bridge across the barriers between disciplines, between departments, which is so essential for this program. The story really started with the late Rabbi Chumimov, unfortunately passed away, and Itzi Parnas were the founder of neuroscience here at the Hebrew University. And then the next step was the formation of the Interdisciplinary Center for Neural Computation, the ICNC, which started as a grassroots operation. Some of the people involved are here, Elon Vadia, Moshe Abeles, who unfortunately left us to another university, Ivan Segev, Chaim Sampolinsky, started as grassroots activity, which then was formalized by creating the center, initially supported by the Orbitz Foundation. It was an initiative of my predecessor, Professor Hanuk Goldfein, who is also professionally connected with the center. It's again a fine example of how a theoretical physicist gets into the problem of understanding the human brain. It's an interdisciplinary or multidisciplinary venture of the best kind of multidisciplinary venture. I will not, so now we are coming to the next stage in the development of brain sciences at Hebrew University. We, about 2 or 3 years ago, as part of the review process of different departments, different programs at the university, we brought a very high-profile committee to the university to study brain research, cognitive sciences, neurosciences, whatever the term you use at Hebrew University. That committee was chaired by Professor Jerry Fischbach, who is here. I'll say something when I introduce him. I'll say something about his wonderful record. Professor Bert Zachman, who is here, was also a member of the committee, very high-profile committee, including some of the top scientists in brain research. And what they came up with was a report that no administration of the university can afford to ignore, which says you've got excellent people doing brain research at the university. You've got even better students studying. I hope that none of the faculty is insulted by saying that the report of the committee was that the students are probably even more promising than the existing faculty. And you should build on that. This is something you cannot, and you should form a really big center in which you bring some of these people together, some of the people will stay in their own departments, invest the resources, even in these difficult times, find the resources, and make it into a great brain sciences center at the university. Fortunately, last week we were able, after long negotiations, got a very important philanthropic foundation, the Safra Foundation, to make a major contribution toward establishing the Edmond and Safra Brain Science Research Center at the university. So it's given me a great pleasure, great satisfaction that we are able to go to jumpstart this very, very important center. And the speakers today will give us a little bit of a glimpse of what is the future of brain research, what are the potential of improving especially today the emphasis will be on medical applications. The other symposium will be with other aspects like the connection between the brain and art. It will be the issues about brain research of the brain and ethical issues of society. And I hope to see you also in the other symposium. I think that we'll start now with Professor El Vadia, who will give a presentation about the basic ideas of the center, the Edmond and Lily Safra Center for Brain Sciences. Edmond is a graduate of Hebrew University, got all his degrees from here, have been since Edmond, didn't have a chance to look at your Edmond. If I remember correctly, since roughly 1980 at the medical school of the Hebrew University Department of Physiology, and been one of the pillars of the ICC, of the Interdisciplinary Center for Neural Computation. His research, he probably will mention it, but his research is about the ways in which the brain controls movement. Errol, please.
This board of governors is really a good excitement and a good opportunity to hear more about the brain, and you will see from what I say that I think this is of utmost importance. I just want to have two brief comments before I start, that this is not the only event about the brain, tomorrow at 9 o'clock we have another event called Brain, Art and Creativity. And on Wednesday we have yet another event starting at 11:30 at the Senate Hall called Neuroscience and Society. I forgot to mention the place of the first one. The first event is at Dan Wasson Auditorium at Yitzhak Rabin Building at Mount Scopus. So, brain, this is the most exciting enigma for mankind since ever, and it's like every mystery, as Albert Einstein says, it's beautiful to study the brain, because the most beautiful thing we can experience is the mysterious. This is the source of all true arts and sciences, and we will hear tomorrow about arts. The challenge of the world is to understand the brain, but we already understand the most important thing about the brain, and this is that our brain is a collection of billions of cells that know how to interact and generate intelligence, emotion, creativity. And we as society must understand this. We must understand that although each of us is an individual with passions and ego and desires, the only way to make achievements is to act as a society synergistically in collaborations, not only in science, in general. And I wish to call from here societies, nations, governments, and leaders to understand how important it is not only to bring peace to this country, but to bring peace to the academic life and understand that through collaboration and through investments like the one Lily Safra well understood and the Safra Foundation well understood, through this kind of investments we open not only to brain research but to understandings among human beings on this planet. And the neuroscientists' challenge is to do their job. And this is our job. We have to do our job, and here at the new center, we reached this point after 15 years of efforts, insisting efforts of all, a large team of people, including at ICNC, but also others at the Hebrew University Administration and in the public. And today we stand in front of a big challenge to make a revolution. We hope to make a revolution, not because we have money, but because we want to find the good ways to study the brain. And we believe that the keys are the following that you see now on the screen. Interactive collaborations, as I mentioned, all human achievements were made by collaborations, not by individuals. The individuals are there to push, but they need to work together like brain cells. We need to look to the future and not to the past. We need to face the future in politics and in science. We need to apply new technologies to study the brain, to study our new ideas. And at this time, I believe there are new ideas about how the brain works, and there are new technologies that will allow us to test these ideas. And key number 3, of course, is to pass this message to the next generation and educate the next generation such that they, I don't consider myself the next generation anymore, that they know how to make the revolution that we hope for. So the next generation. And finally, scientists, neuroscientists, and scientists in general must stop the long-standing tradition, the old tradition that they are in ivory towers speaking a language that no one understands. This is why we have this symposium, and I hope all my colleagues here will speak in a clear language that people can understand. We need to interact with the community, make the community understand that this is the mission of all of us. We do the science, but you need to know what we are doing. And I hope that in this center, and today, we will communicate and interact with people who are not scientists so that they really feel they know what it is all about. It's not just some crazy idea of someone. And this is the key for success of our new center. Now, since the time is short, we are running a bit late, I will have to make the other points that I want to say fast. I think by now I made my message. My message is that through these 4 key aspects, I believe we will change completely what was till now. We will have something completely new, and we hope to participate with all of you in this endeavor. So at the structure of the new center, I will give just a few details, is based on 5 wings, studying the brain from the molecular and cellular level, which is shown here. This is a movie showing that we can go into the cell, look into the molecules, look into the gene, and understand how they encode behavior. The first level is to study the circuits. This is the basic level of computation in the brain. This was Adi Mizrahi of ICNC. And today we can image and see the cells in a living brain. And we see how they change, how they grow, how they develop. We now know that some cells in some areas of the brain are newly born and reach their targets. We start to know more and more about the structure and action of living cells in the brain tissue. What you are seeing here is a brain tissue of living cells. And next, we should have a theory that will allow us to put data into ideas and test new ideas. For example, theory that would allow us to construct cortical column, an operation unit, a local circuit in the brain that operates like the one you see here. This is a computer reconstruction. This is the cortical column in silico, in the computer. Next comes the networks of activities of neurons in this column that we just saw. And what you are seeing now is recordings of real action potential spikes of single cells in the brain that we record, in this case in my lab, and allow ourselves, and this allows us to generate actions by looking at brain activity. Sorry, I lost my... Okay. And next, of course, we want to understand human behavior. Human behavior we can understand through cognition, studying emotion, decisions, etc. One tool that we have today, but is still not right to really understand the brain, and should be developed, should go forward in the functional imaging, like you see here, and taking all these 5 together, all of them, into a melting pot. What we want to achieve is to go in the spirit of this symposium, from brain science to brain repair. We want to pursue therapeutic approaches for restoring normal brain function in relatively young people, 60 and 70, who are sick and really needs our help in the basic research that we do to develop revolutions in science. The revolutions will not come only with the tools we have today. I want to bring you at least 2 examples, or one I will do, of new ways to study the brain that emerges these days. For example, the notion of connectomics, the connectome. Here there is an example of a method that allows us to check for every brain cell, to identify it, to track it, and to see who it connects to. And this is demonstrated in this movie where there is a 3D reconstruction of a single cell with its processes, how it moves, and then with different colors we can see the different cells and see how they connect to each other in this unit that was demonstrated before in silico. And this can be done in longer range, in bigger networks, in long-range connections of the brain. This is the beginning of quantifying the anatomy to the level of the synapse. And this will allow us to understand for real what hardware we have there in the brain, how the nerve cells interact with each other, and how they create behavior. Finally, I will skip one. Finally, I would like to go to the next generation and tell you that at ICNC, and I think at the Hebrew University in general, the next generation was always important. In the new center, we plan to put the strongest stress on the next generation in several ways. First of all, we will recruit new faculty, young faculty, that at least 15 positions we already have, to create a center with interactions of well-experienced scientists and new colleagues that will come to this center and live together in the same place. They will all live in a state-of-the-art new building with core facilities, modern laboratories, where they will do science in optimal surrounding. And we take care of the next generation in developing graduate programs to all of them. By the way, what you are seeing now are students of ICNC with their children. So we are taking care of the students of ICNC, but also of their children as they grow up. And these students will have an international program, which was already announced by the current head of the program, Professor Israel Lelker, that already started at ICNC and will grow with other disciplines of the university to develop and offer advanced courses, home for students where they sit the whole day and study, annual workshop, postdoctoral training abroad, funding for this, hosting postdoctoral students that come from abroad to Israel, and of course the new positions. This is how we look at the next generation. And finally, we should all live in the society and in the community where we are. Our students and us are involved in the community. For example, we give lecture to the public, not only this day, but many times during the year. We participated and put forward an exhibition at the Science Museum where publications we contribute to publications in general reader magazines. We invite people to lectures, and you can read about this in the media. These are lectures for the public, given in a language that everyone can understand. And I invite all of you to continue to follow the publications. We will continue to do this. As these are the list of the lectures in the last 2 years, starting, as you can see, with Bert Zachman as the first speaker, top left, the first speaker of the Heller Lecture Series that we had in the last years. And with this, I want to conclude and tell you that we broaden our mind. We are interested in brain and art, and we are interested in connecting to people. The Brain Circle Club is open for all those who are willing also to invest in it, and it includes today about 40 members, mainly from Europe, and we hope to extend the circle and have an Israeli circle, an American circle, worldwide circle of friends of the brain circle. Thank you very much, and I invite you to join us and see you in the next years as brain science advances here in Jerusalem. Thank you.
Okay, well, the next speaker is getting ready. Let me tell you a little bit about him. We're talking about Professor Bert Zachman, one of the leaders of brain research worldwide. Bert Zachman got his medical degree at Tübingen University. Since then, he's been teaching in Göttingen, Heidelberg, has been the director of the Max Planck Institute for Medical Research in Heidelberg. He is the winner of many, many prizes, recognizing his tremendous contribution to neuroscience, of course culminating in the 1991 Nobel Prize, together with his colleague Erwin Neher, together devoted for developing a new technique for observing or listening to brain, to neural cells called the patchwork clamp. He's a very, very good friend of Israel, of the Hebrew University, And as many people here know, yesterday we recognized his contribution in friendship by awarding him with honorary doctorate of the Hebrew University of Jerusalem. What Professor Brett Sackman will talk about is the future brain research, but mainly concentrating on some of the problems that have to be solved before we can really make use of the knowledge of brain research for medical applications. So really understand it will be the list of problems to be solved. So Bert, thank you.
For introducing me. I was asked to talk about frontiers in brain science, and this is obviously a very subjective affair. What I'm going to tell you is my view of what I think is a frontier, and that can be done in the foreseeable future. Considering my age, I would like to see a few things solved, so I can't have two great visions. Now, the problems I think that are important to solve in my universe is to understand at least part of the cerebral cortex. So if you ask me where is the frontier, it's the gray matter, or gray matters. Now, what is gray matter? The gray matter is responsible for human scientific, artistic, engineering, and also political things you can do. For example, this is Ramon Cajal. Looking through a microscope, he's the father of finding out what are the different cell types in the cortex. He did this with the help of his cortex. This is the Titanic in engineering masterpiece. Before it sank, also it required the cortex. This is a picture by Jean Giraud. It's also part of our cortex. It is necessary for this. And finally, the cortex is also necessary for telling all sorts of stories. This is a German politician telling stories about his past. So the cortex has very different uses, and we want to understand at least part of them. If you want to understand the cortex, the first thing is you have to cut it open, and you find that the cortex is a gray matter. It is a layer of about 5 millimeters in distance, covering the rest of the brain, and we subdivide the cortex or the brain into the gray matter. This is this layer which you see here, and the white matter. Now, from what I said before, most of our abilities that we have depend on the intactness of this gray matter. This gray matter is made up of billions of cells, which we know since more than 100 years through the work of Ramon Cajal. We saw a picture of Ramon Cajal in the second slide, and he found out that the cortex is made up of individual cell types. Mostly of the pyramidal cell type. You see an example here. It's called a pyramidal cell because the pyramid shape of the cell body of the soma. It has another compartment called the dendrite, and another one is the axon. And the flow of information is such that the dendrite receives message and then sends the message via the axon. This is a very simplified picture. Now, also since Ramon Cajal, we know that these millions or billions of cells are connected by so-called synapses. These are the places, thank you. These are those places where these cells are communicating. Now, what I would like to address is 3 different questions that I think might be solvable in the foreseeable future, and I'll go through them sequentially. The first theme would be to identify and understand mechanistically circuits of nerve cells driving a particular behavior. Now again, this can be done in many different behaviors, many different animals, and I'll show you just one example, which is the behavior I'm working on, but this is by no means the only one. You can do the same sort of analysis in flies or in fish and mice, rats, and so on. So I don't want to give you the impression that what I'm going to show you is the preparation. It's just to outline some of the questions that we have to solve in order to understand the basis of a behavior. This slide briefly summarizes what I mean. You define a behavior, for example, whisking with the vibrissae of a rodent. These vibrissae then activate a part of the cortex, which is called an area in a cortical map. In this area, you have a smaller unit. It's called the cortical column. You can see this by this red circle. And this column consists of these pyramidal cells, or mostly of these pyramidal cells, which are connected by synapses. And what we want to find out is how a behavior is driven by the activity of these cells. For this, to answer this question, one has to do two things. First, define the anatomy as accurately as possible, and secondly, to define the activity in these cells that we have identified as being the basis of the behavior. Now I'll go through these two parts sequentially. First, we want to reconstruct the cell populations that participate in a particular population in 3D. I just have to be clear. It's got to be 3D. The brain is a 3D structure, and up to now, most of our anatomy consists of 2D projections, and this loses an important aspect. Therefore, one has to do 3D reconstructions of a cortex, and the first thing is one looks at the number and the location of cells that make up a particular column. The neural cortex has the advantage that we have landmarks by which we can register the data from different experiments. These are the pia mater that's covering the brain, the white matter, you saw that, and in this particular cortex there is a structure called a barrel, which I won't go into. Now, so we have landmarks, and these landmarks allow us to reconstruct cells with a almost unique location or individual location. What you see here is a reconstruction of all the cell bodies in one column. This will be the area of the barrel. Such a column consists of about 15,000 individual neurons, cell bodies. Now, to make this a bit more visual, one also reconstructs the cells around such a column and then uses simple filtering to give you an impression of the differences in the cell density along the vertical and along the horizontal axis. What I want to show you with this is that the cortex is not a homogeneous assembly of cells. It has a very particular structure, both in the vertical and in the horizontal direction. So there are two levels or two planes which are orthogonal to each other. And if you want to understand the cortex, you have to be able to quantify these structures. What you see here is a wider section of the cortex. In the middle, you have this column. These are the individual cells. This will be the barrel. Now, if one looks from top onto this is a top view of this area. One can find again that there is a substructure. If you look at these, and these. So this would be the structure in the horizontal direction. And to show this in 3D, wait a second, there's one missing. Anyway, what you can see here is now a top view. That is, there is a structure in this cortex with different densities along the horizontal plane. So this will be the view on one individual column. This will be the next column, the next column. So what I would like to tell you is there is a very refined structure in this direction and a very refined structure in the horizontal direction. And if you understand the cortex, we have to take care, we have to take notice of the unequal distribution of cells in these two directions. Now, these were just the somata, but what is really important for a neuron are these two other compartments, the dendrite, that's the receiving compartment, and the axon, that's the sending compartment. And again, we have to register those, first reconstruct them and then register those. And I told you we have these landmarks, these barrels, this would be the pia, here would be the white matter, and we can register soma with reference to these landmarks. Then add one structure which is called a dendrite, so this would be the soma, that's the dendrite of this particular cell. This particular cell then is innervated by another cell via axon, so this would be the input axon, and each of these cells has an output axon, which also has a very different structure. So what we want to know is what is the input-output structure of each neuron in a particular part of the brain. Unless we understand this, we're not going to understand the cortex. And I just show you this for this particular neuron. There are techniques available where one can look at tens of cells or even hundreds of cells in the next future, but we have to do this for each particular cell individually, because you can see an axon is very different whether it innervates a neuron or whether it sends away the information from a particular neuron. So the idea is that we have to first quantify the connections between these cells. These cells are connected by so-called synapses. Again, this has to be done in a quantitative way, and I would like to show you this in one particular example. That is a thalamic neuron, which is in the brain stem, or in the midbrain, innervating such a column. This is a superposition of a pyramidal neuron, which you have seen before, and the axonal arbor of a thalamic neuron. You can see that in this particular case, there is a preferential area of putative innervation, which is located in these dendrites. We then have to move from a single neuron reconstruction to multiple neurons. This is shown with 10 pyramidal cells innervated by 10 thalamic neurons, and it's already getting rather uncomprehensible. Therefore, one has to develop methods to quantify the interaction between the innervating axon and the dendrite. And one way that we use and other people are using is to reduce the dimensionality from 3 to 2 and describe the innervation density of such a neuron by way of such innervation density profiles. So this would describe the fact that in this particular case, the highest innervation density, that is the largest overlap between these two cells or cell populations, is located in this area, which is about 1,000 micrometers below the pia. So the future then in this particular aspect of reconstruction is to define average connectivities between individual cell types and quantify them to define a few numbers. Two of the most important numbers are shown here. How many synapses are formed by a particular connection? Usually in the range of between 5 and 10 would be, but it's really variable depending on where you look. And more important is the location of the synapses of a particular connection type. And you don't want to know this only for one neuron, but you want to know it for the whole ensemble of neurons that drive a particular behavior. So this is a challenge for the future, to quantify connections on a cell-specific basis to be able to understand the strength of connections between different parts of the cortex. Now, an equally important question is you have to watch the cells being active when they are involved in a particular behavior. This has been possible during the past 10 years by the invention of two-photon microscopy, mostly by Denk and his collaborators, where one uses an indicator that is brought into these neurons and then can online you an example. So this would be just looking into the cortex. So we are starting from the top of the cortex and image to about 500 micrometers deep in the cortex. And you'll see two colors. One color is reddish. These are glial cells, and the nerve cells are shown in green. So let's just go from the top of the cortex into the cortex. No nerve cells, no nerve cells. Now the nerve cell starts to come. It's full of nerve cells. And at 500 micrometers depth, one doesn't see anything. It's because the image is blurred. Now, can we show this in a somewhat more digestible way? Yes. This is a reconstruction of the film you just saw. So there are cells which you see in yellow. These are glial cells. They are not so interesting for us in the moment. And cells in green, these are the nerve cell bodies. Since all this is done in silico or has been stored in the computer, it can be turned by 90 degrees. Now we can watch a particular layer in the cortex, looking at individual cells. In this case, we can look at about 30 cells at a time. Now, Helmchen developed even a further method that allows us not to look only in 2 dimensions, in a 2-dimensional grid, but also in a 3-dimensional grid. And you will see what is happening in a particular column when a whisker is deflected. The whisker is deflected in order to initiate a behavior. And what you will see is a change of color of a particular cell type. These are the neurons, they are in black. And when the whisker is moved, this is the signal to initiate the behavior. They will change their color, indicating that they're electrically active. Now just watch this, please. Now whisker comes, whisker. Now whisker stimulation is stopped. And what you can see is a brief burst of activity in different parts of such a column. The major questions that arise from this kind of data are, skipping ahead, you can do the same experiment also in the visual cortex, presenting the mouse not with a stimulus that deflects the whisker, but show a particular pattern. In this case, this is a film made by Arthur Connor. The mouse is shown a grating that either is flowing in the vertical direction, in the oblique direction, or in the horizontal direction, and just watch the activity of these different cells. As we move the grating, that's a change of the grating. You can see the vertical, this is the oblique. Other cells, that's the horizontal one. You can see that for each of these directions, a different ensemble of neurons is active. This is shown this is quantified in the next slide. What you see here, we have picked 3 cells. This cell will respond to a grating that is moving in this direction. These 2 cells are activated when the grating is moving in this direction. What this shows is that at least in the mouse there is an intermingling of cells with different selectivity, and it will be the task of the future to find out how the network behaves. What do we want to find out about this? There are a few simple questions. One of them, how many cells are active during a behavior? That is, how many of the available cells? Is it 10%? Is it 100%? Is it 5%? Evidence shows it's about 10%, less or even less than 10%. What is the contribution of the different cell types? Is a behavior driven by a particular cell type? Not all cells are equal. I showed you one type, but there is good evidence that different cells participate in different behaviors. What is the degree of synchronization between cells during behavior? You could have seen this in the last movie, that 2 of these cells are almost active at the same time. And the next question is, can synchronization between cells lead to the learning of a behavior? These are very simple questions. We have the data now. What is lacking is proper analysis methods, and I'm very hopeful that the new Safra Center will be one spot in the world where the knowledge to analyze such kind of data is accumulating, and I'm very much probably know that Alzheimer's disease is associated with the appearance of plaques. It's not quite clear how these plaques are affecting the neural network at an early stage, but there are two recent papers that have really made a big step forward, which I would like to show. One is by the group of Hyman at Harvard. What they showed is that such a plaque that which is shown here in blue, these are neurites, dendrites, and axons from nerve cells, can develop sort of daughter plaques within a relatively short time period. If you focus on this, then you focus on this, and you focus on this. This is a time period of about 24 hours. So within 24 hours, there is a new plaque coming up, and they also showed that around this plaque, there is a very strong damage in the structure of such a cell, which also occurs in a very short time period. So, contrary to current belief, such changes, that is the appearance of plaques and the damage caused by substances emanating from these plaques, occur in a very short time range comparatively, and we will be able to use this to look at the very initial stages of damage caused by these Alzheimer plaques. Even more exciting is a finding by Conrad and his group showing that around the plaque, this again will be the blue structure, there are two types of cells. These are the neurons, these are the glial cells, and what they showed is that before there is an anatomical change in these cells, there is already a change in the relative activity of these cells. Some of the cells become more active, some cells become silent, and other cells do not change, and it's not quite clear what is happening, how this depends on the vicinity of the plaque, but it is a means to find out at the very early stages what is happening around an Alzheimer plaque before it has developed. So this is an area which has been enabled by the development of tools that were developed for completely different purposes, but it's a typical example of how tools in biology suddenly can be useful to identify or to find out about the cause of a particular disease. So the final frontier in my thinking is that we have to understand brain activity in humans. You know these pictures. This is a technique called functional MRI that we must have seen, where the cortex is showing up in these blobs, depending on the activity, as it's called. It's more active or less active or inactive. But one has to realize that fMRI is basically a blood flow signal. We are looking at the blood flow. We are not looking at the electrical activity. Also, this is very often mixed. And one way to do this is to measure at the same time the blood flow and the electrical signal and the fMRI signals. And there is really pioneering work by David Kleinfeld in California, who recently managed to reconstruct the vascular pattern in such a column or in a number of columns, and he also measured the blood flow related to the electrical activity. And the message is they are not strictly correlated. So one has to be very careful in interpreting these results. And one of the frontiers in this area will be to find out what the fMRI signals are affecting. Unless we know what is reflecting, we cannot interpret. We can't just make more and more landscapes with colorful blobs, but he's not telling us what's going on. And I think this is one of the most important areas if one wants to relate basic science to, let's say, what's going on in the brain. So I had the chance during one of the dinners to talk to one of the students, and maybe you have seen through my lecture that I think anatomy is a really important field. There is a saying, if you don't understand the function, do the structure. simulate nerve sets on the basis of rock-solid data, and anatomy is rock-solid. So I think this is a message to those who run this institute. My hope is that something similar will develop out of this center. You know, Plato's Academia had as a motto, let no one ignorant of geometry enter this building. And I just wonder whether we could, or you could invent a motto for the new Safra Center. No one ignorant of detailed anatomy should be allowed to work in this. This is very, let's say, provocative, but at least it gives you some thought. And possibly, the Safra Center in 2,000 years could be regarded as Plato's Academia that we look upon now. Finally, for the Safra Center, I would say Mazal Tov, and I also would like to say Mia V'Isrim. I was told Mia V'Isrim could also mean we need altogether $120 million to get this.
Okay, our next speaker is Professor Gerald Fischbach. Jerry, I guess by now we have very close friends. We call him Jerry. Got his medical degree from Cornell. Since then, he's been one of the most prominent neuroscientists in the United States. He's been teaching at Washington University, at the other HU, namely Harvard University. And he's been the head of for 6 years, the head of the National Institute for Neurological, what's the official title? Neurological Disorders and Stroke, part of the NIH. And he's been the Vice President for Health Sciences and the Dean of the Medical School at Columbia, which he retired about a year ago. The last couple of years, he was very famous for dealing with the synapses, the connection between the axon and dendrites. You see, I'm becoming a brain scientist myself. The synapses, the connection, the mechanism behind the connection. But the last couple of years, his main interest is the psychological mechanism behind a very terrible disorder, which is autism. And since I know many families with children, autistic children, I know how terrible that could be for a family. So if that could give any hope of dealing with autism, I think it could be a major contribution to humanity.
Thank you very much, Malcolm, and thank you for inviting me to come and attend this very important meeting of the Board of Governors. I'm going to talk for 15 minutes about autism. I have studied synapses all my career, and yet in the last 3 years, I've turned to a study of autism because I feel in many ways autism and diseases related to autism, neuropsychiatric disorders, are the culmination of many things that we work for. And I think it, in many ways, is the culmination of what the Safra Center for Neuroscience can grow into and will benefit from, and in turn will influence the field. Many people have told me, Jerry, at your age, it's a privilege to have a steep learning curve. And believe me, this has been a steep learning curve. It's been actually a wonderful experience coming to learn more about this disorder. Now, autism was described in 1943 by Leo Kanner, and he knew that Donald T. was not the usual child. He knew that as soon as he read the letter from Donald's father, which described the boy as being happiest when he was alone, drawing into a shell with himself, oblivious to everything around him. His father said that Donald had a mania for spinning toys. He liked to shake his head from side to side and spin around in circles. And he had temper tantrums whenever his routine was interrupted. And these characteristics of aloneness and an obsession with preservation of sameness are the hallmarks which have stood up since Leo Kanner's description in 1943. There are also language delays. The aloneness is reflected in a certain lack of understanding of other people's beliefs, a lack of joint attention and play, a lack of empathy. Autism is usually diagnosed by age 3. The definition of autism has grown at both ends. At the high end of autism, some people describe as Asperger's syndrome, and the low end it blends into mental retardation. In my view, the expansion of the definition of autism is responsible in large part for the reported increase in prevalence over the years, which today has reached the astounding number of 1 in about 150 births. Boys are 4 to 8 times more likely to become autistic than girls, and it shows no racial or geographic boundaries. It's a major, major public health problem. It's not a disease at the end of life, it's a disease at the beginning of life, and it is with us for generations. It's with the affected individual, with their families, and with society. So I am now at the Simons Foundation directing their autism research initiative. And our overall goal is, as said here, to support fundamental research. We're not involved yet in clinical research or in policy matters, but we support fundamental research that will lead to a deeper understanding of autism and along the way of the human mind. And I'll try and point out towards the end what I mean by that. Now, what does it mean to understand autism? We've taken several different approaches, and this was reflected in Elon's introduction. We are taking the approach of bottom-up, looking for molecules and moving to mind by studying gene identification, and then once these genes are identified, to understand what they do, how they affect nerve cells and synapses, and then to understand how these nerve cells and synapses focus in neural circuits, both small circuits, local circuits, as described by Bert Sockman, and larger neural systems, and how these neural circuits, small and large, contribute to our understanding of the behaviors that are described as autistic. Now, there's no question that autism is a genetic disorder. One simple fact will tell you that the correspondence in identical twins between one twin who is autistic, and the likelihood of the second twin developing the same symptomatology in some studies is 90%. And that's remarkable. It's the highest percentage I know for any neurologic or psychiatric disorder. Genes must be involved, but they're not involved in the usual way, the way you may think. And I'll give you examples of each one of these approaches. We're funding now, by the way, more than 90 investigators, and we are evaluating families at 13 sites around North America, the U.S. and Canada, in an attempt to identify genes. The genes are identified in various ways, by studying families, linkage analysis, by GWAS, genome-wide association studies, looking at large populations, 2,000 to 50,000 people, by looking for de novo mutations, and this is what I'm going to dwell individuals with autism usually don't marry and don't have children. So how can this be a genetic disorder? It's not a disorder that's passed on from generation to generation. It's now thought, and we are acting on the assumption, that these are new gene mutations that happen in every generation. And they probably occur in the germ cells, in the sperm of the fathers and the eggs of the mother. A study consistent with that was done here in Israel, studying individuals in the Israeli army over 6 years, studying their medical records. And over 300,000 individuals were involved. And it was found that fathers over the age of 40 were almost 6 times more likely to give rise to offspring who were autistic than fathers under the age of 30. This was a landmark study that has influenced, has influenced the entire field. And it's consistent with the notion that there are mutations that occur in the germ cells as parents age. In this case, the study was focused on fathers. So I will give you one bit of science and talk about what a de novo mutation looks like. The advance here has been rapid. The disorder was described in 1943. Of course, there were autistic individuals before then, but they were given pejorative names such as imbeciles or dim-witted or weird or retarded. But the term autism was in 1943. But there's been enormous progress in the last 10 years, much of it in genetics in the last 3 years, given the increasing power of the technology. So we will talk about de novo mutations. Now this is if I can point here, a genome scan along one particular chromosome, one of the 23 chromosomes, chromosome number 16. And the DNA has been chopped up into pieces. And each piece has been compared with normal control subjects. And ordinarily, we have 2 copies of each gene, one from your mother and one from your father. But in some cases of autism, there is only half the number of genes. And this extends over a pretty large piece of DNA. There are 29 genes in this segment. This is called a copy number variant, and it is a deletion. So this particular individual is walking around missing one copy of 29 genes and was autistic. Interestingly, there are other people in the population who have more than 2 copies of a gene. This individual has 3 copies. And this individual, a duplication of the same segment where the deletion led to autism, was diagnosed as schizophrenic. An interesting coincidence, but maybe not a coincidence. It was thought for a long time that autism was a childhood form of schizophrenia. I don't believe that, but the genetics are leading back to that notion. Indeed, the word autism comes from the schizophrenia literature. Ernst Bleuler first used the term to describe the introversion and the isolation from the world of individuals with schizophrenia. Now, I will say a bit about molecular mechanisms and then move on. I have studied synapses throughout my career, and as Bert Sockman implied, the business of the nervous system, the communication between cells, which must be disrupted in some way in neuropsychiatric disorders, occurs at synapses. And this is a cartoon of the nerve terminal ending on a synaptic target, and these are vesicles packed with neurotransmitter, and when the proper signal arrives at the end of that nerve terminal, it releases the transmitter and affects, either excites or inhibits the next nerve cell in line. And our assumption is there's something amiss about synaptic transmission in the disorders like autism. And indeed, although I want to point out to you, you can't plan anything, there's been an increasingly productive identification of genes that code proteins that act at synapses. This is an example of a story that began almost 15 years ago with the study of black widow spider web. I use it to illustrate that try as we might, we cannot plan for every step in progress. There is a role for serendipity, for brilliant research that initially seems disinterested, but will turn up molecules and ideas that will be useful later. This story originates with a black widow spider venom and how this spider can kill an individual. It does it by releasing a toxin that is injected into the victim's bloodstream and that causes immediate paralysis. Excuse me, here's the picture of the black widow spider venom releasing the transmitter. And in short order, the target of that toxin was identified, the protein purified and crystallized, and it was given a name called nerexin. And its partner in the postsynaptic cell was given a name called neuroligin. And the reason I use this example is that the nerexin gene and the neuroligin gene have both been implicated in autism. Indeed, in the last this is cycling in the wrong way. That ain't me. I'm going to stop here. In the last several years, a number of genes and their protein products have been identified as synapses. And in the last 3 years, these different genes have turned up in genetic studies of autism. So all arrows are pointing to the synapse as a target. Now this is just in the receiving end, the postsynaptic side. There will be many more autism risk factors that affect the presynaptic side, the side that releases the transmitter, that brings the signal to the synapse. Our bet is that there will be more than 100 autism genetic risk factors identified in the next 2 years. And when we have those in hand, we'll be able to understand environmental influences much better. And we'll be able to move on to studies of neural systems and neural circuits. And I'll say just one word about the neural systems. Interest is focused deep in the middle part of the brain. And this is looking at it from the inside out. And in the anterior region, the anterior lobe, sometimes called the prefrontal cortex, seems to be a critical region of the social brain, the part of the nervous system that is active in understanding other people's beliefs, in making decisions about other people, and in other forms of focused attention and ability to allocate attention to individuals. And it is our job to understand what about the local circuits in this area, using those gene products, might lead to altered behavior that could lead to autism. And Bert, I would add a third dimension to your three-dimension anatomy, and that is time. You have to understand how these neural circuits develop initially, how they mature, and how they age. So once those wonderful computer graphic approaches are understood, and once we have the genes in hand, we'll have to understand where in the brain they're active, what they do to these neural circuits, and when they act. And for that, it's going to take a kind of computational science that must be developed in parallel. Now, looking ahead as I'm going to end this talk, there are reasons for hope, and I believe in studies of disorders like autism. One is the scientific method, and I'll end with that. One are advances in the areas we've talked about today. One is the desperate need. These are not just diseases of individuals, but disorders that affect our society in a damaging way, not only economically, but socially. And there's an enormous desire to address these issues, and there's great talent. The reason I'm going through that is I believe that Hebrew University is one of the 5 to 10 universities in the world that can make advances at each one of these levels of analysis. They will, as everyone will, face challenges. This is a complex system. New thoughts are needed, new breakthroughs, new ways of looking at things must complicate the technology, and that's hard to predict. There are challenges in the conduct of science and the type of cooperativity that's going to be needed. The old Robinson-Crusoe model of one person working in her or his lab alone is not going to suffice. There's a desperate need for money, here and abroad. And there's a need for understanding and trust and respect on the part of the public to support this effort. And all this is occurring at a time, at least in the States, and I know around the world, that was severely disrupted by 9/11. Now, it was with this in mind that I really was thankful for the opportunity to come and share the committee here that led to the recommendation of what now is the Safra Neuroscience Institute. And we recommended several things. Bert was part of that committee, and it was a very distinguished committee. We recommended that an institute be formed, an umbrella organization that would bring together components of the entire university. And I do believe in the quote from that report, that great universities, including Hebrew University, will be judged by how it contributes to an understanding of the human mind in the next decades. And understanding pathophysiology is one route to that effort. We recommended a new building be constructed to house new recruits that will be used to rejuvenate the faculty and that can serve as a center of gravity for the entire university. We recommended that a research endowment be created that would aid and help make the members of the institute somewhat independent of external funding. And we recommended the institute become intimately involved in the education mission of the university. Now, I believe I will be most satisfied when we understand the neural circuits that give rise to behaviors, as Bert was discussing. And I believe that the group here at Hebrew University is among the premier places in the world to synthesize this information and develop new theoretical approaches that are needed to penetrate this complexity. And I believe that Hebrew University is situated in the world in a place that can really have a huge impact. And I will end by talking about the values of the scientific method for our society. The objectivity, the sharing, the democratic nature of science, the humanistic approach, and the constant need and desire for more education. And I can illustrate that with one of my favorite examples. This was a paper in my last subject, my last of the talk, published in 1999, which represented a major breakthrough in the area of developmental disorders. It was the discovery of the gene that causes 95% of the cases of Rett syndrome, R-E-T-T-S. This is a disorder of young girls who develop normally for 2 years and then go into a horrible decline. And it is related to autism in certain ways. It's part of the autism spectrum disorders, according to the Diagnostic and Statistical Manual of Psychiatrists. And it gives us hope. It is a single gene disorder. It's not as complicated as autism, which is a multi-gene disorder. But the things I want to point out here are not the science. The senior author here was Huda Zogby, who works now at Baylor University in Houston. She was brought up in Lebanon, and went to school at the American University of Beirut and suffered during the tremendous period of disruption in Lebanon through the 70s and 80s and came to Baylor. And she teamed up in Texas with Ruthie Amir, an MD student from Israel who came to Baylor and Houston with no scientific training, but she came for personal reasons to accompany her husband. And Huda and Ruthie became close friends. Huda taught Ruthie everything she now knows about science, and together within a few years published this remarkable paper. And to me it's inspiring that the science overcame whatever political difficulties they may have had a priori. And to point out the success of this, I know for every scientist in the audience, this is meaningful. This paper was submitted on August 26th and accepted September 1st. That has to be a world's record for acceptance and for the impact that this paper has had over the years. And I think it's a role model. Ruthie came back to Israel, studied with Aaron Shekanover, and is now an independent scientist here. And it's my hope that the Safra Center will use this as a role model, will reach out across the world, and will spread its efforts in brain science and influence us all. Thanks.
So you see, when I tell you about the potential of the Safra, Edmond and Lily Safra Center for Brain Sciences, I do have some grounds for believing in the future. Our next speaker is one of our own, though Bert and Jerry, I think you can consider one for our own. Professor Marta Razin-Weinstock. Marta got her education in England, Chelsea College of Pharmacy, and got a PhD from St. Mary Hospital Medical School. Moved to Israel in '69 to another place, Tel Aviv University. I think in '79 she made the right move. Moved to the Hebrew University School of Pharmacy, and she's dealing with the pharmacology of the nervous system or brain system. She was in charge of developing several drugs, in particular a drug for Alzheimer. Unfortunately doesn't cure it, but slows it down. A drug which is a big moneymaker for the Hebrew University called Exelon. So she's a great contributor to the university. But she's got much more ambitious goal now, which Jerry was talking about at the beginning of disorders of beginning of life. Now we're talking about disorders at the other end, namely the deterioration of the brain due to age. And Barbara's got the ambition that we can do something about deterioration of the brain at older age. This is something that definitely, I guess that I and many others are very much hoping that we'll make great progress on this project. Thank you very much, Marta.
Thank you, Menachem, and thank you for inviting me. I'm very grateful to the two previous speakers because I hope it will make my task a little easier. As you see, the title is Advances in the Treatment of Alzheimer's, and towards the second half I'm going to talk about a new approach that we're trying in my lab. It's a little way out, but I hope I'll be able to convince you. For those of you who are not yet familiar with this devastating disease, problems. Thank you. This one. Alzheimer's disease is the most common form of dementia in the elderly, affecting about 5% of the population worldwide over the age of 65, and it increases to more than 25% at the age of 85. There are those who put that figure even as high as 40%. Now, Alzheimer's disease is a progressive degenerative disease, and it has several causative factors, and we have to remember that when we're trying to address possible ways of treating it. The most characteristic early finding is a loss of short-term memory. We all know people who start talking to you and they'll forget in the middle of a sentence what they're going to say, and they can't remember what they were told a few minutes before. And this, as it progresses, develops into impaired cognitive function, impaired executive function, that's what's called activities of daily living, and most distressing for the family are also behavioral abnormalities. Now that you've heard about the brain and neurotransmitters, I'm just going to show you what we knew about Alzheimer's disease when I started my research in this subject in the 1980s. We knew that these neurons here, which release a chemical transmitter called acetylcholine, were degenerating. Now, this information we achieved or we obtained from post-mortem studies. That means this was pretty late on in the development of the disease, but what was found was as these neurons degenerated, they released much less acetylcholine, and this was related to the degree of memory impairment. Just to show you what's going on at the end of one of these nerve terminals, here you see the vesicles, which we saw in a previous lecture, which contain acetylcholine, and the problem was that not enough of this acetylcholine was released to interact with its receptors on the next neuron. The idea I had at that time was to prepare something which will inhibit this enzyme called acetylcholinesterase, whose function is to destroy acetylcholine immediately after it's activated its receptors. By inhibiting this enzyme, we will allow more acetylcholine to be present in this gap and therefore increase the chance of activating the receptors. There are now at least 3 cholinesterase inhibitors on the market: donepezil, rivastigmine, and galantamine. We synthesized and tested rivastigmine, which is called Exelon, in 1984, and it was subsequently developed and marketed by Novartis. Since then, we've learned quite a lot about acetylcholinesterase inhibitors. They've been on the market for more than 10 years, and we now know that they improve memory, particularly attention and cognitive function, in a significant proportion of patients for up to 2 years. Unfortunately, there are still a large proportion of patients who do not derive benefit from these drugs, but at the moment, there's nothing else. Also, as far as we can tell, they do not significantly affect the progress of the neuronal loss and brain shrinkage. This is a summary of the experiments that have been done with Exelon, rivastigmine, and here you see, here's a measure of cognitive function, the ADAS-Cog. Here are patients who were given placebo, and you see the typical decline in cognitive function over a year. It goes on and on. And for a period of time, rivastigmine and the other cholinesterase inhibitors seem to be able to delay this progression, but eventually it goes on. Now, what are the characteristic changes in the brain of Alzheimer's disease, in addition to what I've just shown you? We heard before that there are extracellular plaques containing beta amyloid. This is a very clear hallmark of Alzheimer's disease, which is seen in most patients at post-mortem. But there are also tangles, neurofibrillary tangles, which are found inside the neurons and significantly disrupt neuronal function. So the plaques are outside, the tangles are inside. And then what we see quite clearly is this progressive brain shrinkage. Here's a healthy brain, here's an Alzheimer brain. It looks rather like a nice juicy plum turning into a prune due to this extensive neuronal loss. Now, as Professor Sackman mentioned, quite a lot of emphasis has been placed in the last few years on these beta amyloid plaques. And an enormous amount of money has been spent in trying to find ways of getting rid of these plaques, whether it's by immunization or preventing their formation. Unfortunately, these treatments which have been tried in patients have not had any significant outcome on cognitive function, even though many of the plaques disappear. Now, it may be that there's something else wrong with the treatments, it's early days yet, or it may be because tangles, which are definitely present and cause a lot of damage, are not affected by the treatments directed against beta amyloid. Or, my guess is that by the time we treat Alzheimer patients, whether with a cholinesterase inhibitor or something against beta amyloid, it is too late. By the time the patient comes to the clinic and the doctor makes the diagnosis that he's definitely got Alzheimer's disease, he has lost far too many neurons and a lot of damage has been done. And if we're looking to the future, I really think we're going to have to pin our sights on something else and start much earlier. And some of the early, so in order to do this, and in my research, I have focused on what is known now about early changes that occur in the aging brain before the presence of plaques and even tangles. And this you see in an aging healthy brain, and it doesn't matter if it's humans, rats, or mice. And there you see evidence of what is called oxidative stress. This is due to the production of free radicals, and these occur particularly in the brain because of the enormous oxygen requirements of the brain. And these free radicals live for an extremely short time, but long enough to damage cell constituents, components of the neurons, leading to their loss. The other thing that one can see in the aging brain is the activation of glia. We saw pictures of astrocytes and microglia before. These are non-neuronal cells that were thought for many years just to be a sort of packing between neuronal cells, but it's quite clear they have very distinct, important functions in the brain. And under certain circumstances, particularly in association with nerve damage, they can release nitric oxide and together with potentially damaging chemicals called cytokines. Now, just for those of you who are not familiar with the chemistry of oxidative stress, oxygen, when it loses an electron, becomes a superoxide anion, which under normal circumstances is converted extremely rapidly to hydrogen peroxide by superoxide dismutase, and then by catalase it is converted to water, so everything's fine. In addition, during a normal neuronal activity, and particularly after activation of microglia, another substance is released, nitric oxide, which comes from the amino acid arginine. And this, together with superoxide, forms the peroxynitrite radical. Now we can show that this is present because this attacks amino acids like tyrosine, and by using suitable antibodies against nitrotyrosine, we can detect its presence. What happens in the aging brain is both superoxide dismutase and catalase become less active, and therefore we are losing some of the mechanisms which help us not to lose brain tissue. Now, as I say, there is now plenty of evidence of oxidative stress in elderly subjects, particularly if they already have mild cognitive impairment before they develop Alzheimer's disease, or even before that. And this is a study done by Practico and his group, where they used a specific marker of oxidative stress, and they measured in the urine, the plasma, and the cerebrospinal fluid of patients. They didn't have to wait till they were dead. And you see here, each one of these dots represents a patient. And you see, if you compare those with Alzheimer's disease to the matched normal controls, there's a larger number of these with evidence of oxidative stress, but already in mild cognitive impairment, there is almost as much of this oxidized protein as there are in Alzheimer's disease. So this is a very clear precursor of what is to come. Now, in order to address these problems, several years ago, we designed a new drug, which was called ladostigil. It inhibits acetylcholinesterase like ribostigmine, because we decided whatever happens, it can't be bad, to support the acetylcholine, which will try and help its activity. In addition, it inhibits another enzyme called monoamine oxidase, and monoamine oxidase is responsible for the breakdown of other neurotransmitters, particularly dopamine and noradrenaline, which are very important because they are also lost in Alzheimer's disease. And their loss may contribute to some of the behavioral problems, particularly the depression. And in the course of working with this drug, I took a chance and I started to look for different mechanisms unrelated to the ability of the drug to inhibit acetylcholinesterase or monoamine oxidase. And I discovered that indeed this drug has neuroprotective activity against both oxidative stress and glial activation. And this is just one of the many experiments we did using confocal microscopy. And here you see cells which are normal, and the red dots here show the mitochondria. Now, another thing that happens in the aging brain is a loss of mitochondrial function. And of course, once your mitochondria, which are the sort of powerhouse of the brain, begins to lose some of its activity, then there's a problem with neuronal signaling. Here you see normal cells, nice red mitochondria, but when we induce oxidative stress, you see the red changes to green because of a fall in the mitochondrial potential. If we pre-treat or co-treat the cells with ladostigil, we essentially prevent the fall in this mitochondrial potential. In other words, we have prevented the effect of oxidative stress and cell damage. And in another study, we took isolated microglial cells and we activated them with a bacterial toxin called lipopolysaccharide, and under these conditions, the cells release nitric oxide, as I showed you, and two cytokines, IL-1 beta, TNF-alpha, and at a concentration of less than 10 nanomoles, which is very little, our drug inhibits, here by 25% and more, the release of these cytokines from cells. Of course, that's all very well working in the test tube, but there comes a time when you really have to show whether the drug can work, at least in the animal. And our results were based on a rat model, which shows these early changing in the brain as they age. And now a significant proportion of old rats develop memory deficits, particularly spatial memory, which is the easiest thing to test in rats, but they do not have plaques or tangles. But the brains show evidence of microglial and astrocyte activation and of oxidative stress. In our studies, in the strain of rat we used, at least 60% of them develop memory deficits when they're aged 22 months. When they're 16 months, and most of them are still all right, we gave them every day a low dose of ladostigil, 1 milligram per kilo, and kept them going until they were 22 months. Now, most of these rats die when they're about 24 months. And we sectioned the brains, and what I want to show you here are a large increase in astrocytes, active astrocytes. You see the red cells here with lots and lots of branches compared to the young animals. And when these animals had been treated for 6 months with ladostigil, there were far fewer of these active astrocytes. And here you see a quantification of this. Note also that when young animals, from the age of 3 months, were given ladostigil, nothing happened to the astrocyte population, but in the old ones they were reduced right down to the level of those in young rats. And similarly, microglia. Microglia is where many of the potentially damaging cytokines come from, although cytokines also have very important uses. But you see here, different areas in the hippocampus. I've just taken examples. This is a white matter area, this is a parietal cortex. You notice immediately that all these areas in the aging rat had much more, or many more, activated microglia than the young rats, and this was reduced right back down to the normal level by our drug. And here, just to show you a bit, when I say activated microglia, in the previous slide, that was done by means of an antibody which specifically attaches to activated microglia. But here you see they actually change their shape. If we look at normal microglia, they're sort of thin structures with some branches, but here they become absolutely rounded and macrophage-like, which is really a sign that they're ready to go and do something in the brain. And here again, those who were treated with ladostigil had far fewer of these rounded macrophage-like cells, as you see in this quantification. The other substance that we measured was a precursor of the nerve growth factor, pro-NGF. In aging, it is known that there is a decrease in neurotrophic support. This is one of the reasons, possibly, that we're losing our cholinergic input and others. And this is shown in a specific hippocampal region by the loss, as I say, of the NGF precursor, pro-NGF. And here, I hope you can see this better than I can, pro-NGF is shown in green, all these little green dots. This is a specific area of the hippocampus, which is very important in rats for spatial memory. You see in the aged control, we virtually lost our pro-NGF staining. You can see again the red astrocytes, which are far more numerous here than here. And again, when the rat was treated with ladostigil, the green staining comes back and the red astrocytes are lost, as I showed you before. And here it's quantified. We didn't get it right the way back, but it's quite clear there's a huge difference, and this is definitely on the way. Now, how did all this affect the memory of the rat? Well, we test memory in rats at various procedures, one of which is to put him in a large circular pool 1.6 meters in diameter, and he has to find a submerged platform. They don't like going in the pool, particularly if the water's quite cold, and they make quite heroic attempts to find this platform. And every day we change the position of the entry of the rat, and we leave the platform in the same place. And what you see here in the young control, from trial to trial they get better. In other words, they take less time to find that platform until by the 4th or 5th trial they go straight for it. However, if we look at old rats, rather like old people, they have good days and not such good days. You see here they took much longer to get started, then they seem to be all right, oops, back again. This is very, very typical of the behavior of an aging rat, and those which were treated with ladostigil behave essentially like the young ones. So to summarize the neuroprotective actions of ladostigil, we can say that ladostigil prevents the effects of oxidative stress, the fall in the mitochondrial potential that I showed you, cell death measured by other means, the release of nitric oxide and cytokines from activated glia. When given chronically to rats, ladostigil prevents glial activation, neurotrophic loss, and the development of spatial memory deficits seen in old age. The drug has been tested for safety in patients, and it is extremely safe compared to other cholinesterase inhibitors. What we're now trying to do is to recruit enough money to run a phase 2 efficacy study in which we would use the normal doses just to see if it is better than existing cholinesterase inhibitors, because it also inhibits monoamine oxidase. However, and this is more important, in an independent trial, we would like to test the potential effect of this drug on neurodegeneration, in which we will give a much lower dose, and we'll have to give it for a minimum of 2 years, and see what happens in a whole battery of neuropsychological tests, together with repeated fMRI or other measurements, which will show us whether, in fact, the drug is able to have any impact on brain shrinkage. And hopefully, if it can have some effect on this reduction in key brain areas, it may eventually be useful in reducing the rate of development of Alzheimer's disease. I'd just like to mention my colleagues and students who have helped me in this research endeavour, and that the beginning of our research was supported by Teva Israel.
Thank you very much, Marta. Maybe rather than looking for the fountain of youth, maybe look for the peel of youth. Our next speaker is Hagai Bergman from the Hebrew University Hassan Medical School. Hagai got his medical degree from Technion. Nobody's perfect. And then went for postdoctorate studies with Moshe Aveles at the Hebrew University. Then went to Johns Hopkins and came back as a faculty member to the Hebrew University Medical School. Hagai, besides other things, deals with Parkinson's disease. He's one of the leaders in dealing with deep brain stimulation treatment for Parkinson. He's got some novel ideas of how to improve in a substantial way this technique. So it's definitely a terrible disease, and if you can make any contribution to dealing with it, it will be a great benefit to humanity.
Thank you, Menachem, and thank everybody for coming. And as Menachem said, I will speak about deep brain stimulation for Parkinson's disease in the past, present, and the future of this treatment. Parkinson's disease is the second most common neurodegenerative disease after Alzheimer's disease. It was initially described almost 200 years ago by James Parkinson, and the disease is composed mainly by lack of voluntary movement of the patient. And what we see is that the patient is losing his motor vigor. And over here you may be able to see one of our patients, and we see this patient in 3 movies. The patient agrees to be video and to show symptoms to us. And you may see the lack of speed of movement, the mask face, and the akinesia, the loss of motor vigor, which is one of the main symptoms of Parkinson's disease, in addition to other symptoms like tremor, muscular rigidity, and other symptoms. Parkinson's disease, as we said, is very, very common. It is 1% of the population above 60, and today we know that it's caused by the degeneration of dopaminergic neuron located at the midbrain and projected the neurotransmitter dopamine up to the input stage of the basal ganglia, the striatum. This is in the normal, but in the Parkinsonian brain, we see that there is much less dopaminergic neuron in the midbrain and no dopamine in the striatum. So, having this in mind, it took a little bit, about 150 years, but in 1970, actually, it was discovered that we can treat our Parkinsonian patient by giving them back their dopamine. We cannot give dopamine, we have to give L-DOPA, which crossed the blood-brain barrier, but since the introduction of the levodopa treatment, that was beautifully described by the book of Oliver Sacks and the movies, then we know since then and up to today, dopamine replacement therapy is the main core of our treatment of Parkinsonian patients. And indeed, the beginning is very, very good. We can treat our Parkinsonian patient, and for 5-10 years, everything is very good. But as you may see, as time passed from the beginning of the treatment, so after 10 years from the beginning of treatment, about 100% of our patients develop side effects of the treatment. And these side effects include, so this is the same patient that we see before, but now after dopamine replacement therapy. What we are getting is a situation that we call dyskinesia, too much uncontrolled movement. And we cannot help the patient because what happened with time is that the therapeutic window that was very, very good in the beginning, below no effect, above too much dyskinetic effect, after 10 years of treatment on average, the therapeutic window became very, very small, and our patient either cannot benefit from the drugs and is akinetic, or have too much and have dyskinetic disorder. So the next step in our understanding of Parkinson's disease happened about 20 years ago. This happened in California when a group of young people have tried to synthesize synthetic heroin. And unfortunately for these young addicts, inside this synthetic heroin, there was a trace of neurotoxin called MPTP. And when they injected the synthetic heroin and the MPTP into their blood, they developed all the symptoms of Parkinson's disease due to degeneration of their dopaminergic neuron. This gave us the miracle that we can now have an animal model for the disease. And if we inject MPTP to monkey, this monkey mimic all the symptoms of Parkinson's disease, and therefore we are able to record the electrical activity in the brain of this monkey and see the electrical activity in the normal monkey. This is the normal monkey, and this is what is going on after induction of Parkinson's disease in the monkey. And what we found is that in some places of the basal ganglia, there is overactivity. The number of action potential emitted by the neuron in a small area of the basal ganglia, the subthalamic nucleus, is much higher than in the normal monkey. And therefore, we decided in 1990 with Thomas Wickman and Edmond to inactivate this small structure of the basal ganglia. And indeed, what happened is that if we look at the number of movements, so this is the subthalamic nucleus, and we are looking at the number of movements the normal monkey is doing, and the monkey is doing movement, then after MPTP and the development of Parkinsonian symptoms, the monkey is akinetic, much less movement, but after inactivation of the subthalamic nucleus in this monkey, either by muscimol or botanic acid, the number of movements became equal to the number of normal movements. Very similar for rigidity and for tremor, no tremor in the normal monkey, and lot of tremor in the after MPTP, but again, a significant reduction in the amount of tremor after inactivation of the subthalamic nucleus in this monkey. This idea was immediately taken to the operating room. 3 years later, in 1993, Professor Benabid from Grenoble took it to the human operation, and since then, all over the world, there have been more than 40,000 operations, and it is included in the national program of our country, and since 4 years ago, we've done more than 150 patients in Hadassah Hospital. What we are doing is that we have a very, very sophisticated imaging system. With MRI, we locate this small structure in the brain. We put a frame, a metal frame on the skull of the patient. We make a small hole in the skull, and then when the patient is awake, we put our electrode, the same as we've done before, into the monkey, find the electrical signature of the subthalamic nucleus, and implant over there an electrode which is going to internal pulse generator located at the chest of the patient, and using this pulse generator, inactivate the subthalamic nucleus. And now you will see the same patient as we did see before, and this is after the implantation of the internal pulse generator into the subthalamic nucleus, and as you may see, the patient is moving, is smiling, he got back his motor vigor. And as I said, since 1993, more than 30,000 or 40,000 operations all over the world, and 150 patients in Hadassah. What are the next stage? Clearly, in order to understand better, we have to understand better the situation. And the first thing is to understand the normal function of the basal ganglia and the dopamine system. And this is a study that will be published this week, this Thursday, in Neuron. The study was mainly done by these students, Avital Adler and Matthew Joshua. Joshua. So this is my gift to the board of the university, a paper neuron that show that dopamine neuron encode events that are better than predicted by synchronizing, by changing their amount of synchronization. So they don't only encode the behavior of the events by changing their firing rate and firing pattern, they change their synchronization level as well. Secondly, we have to look at our patient. And what we are doing is that we are using the data that we are recording in the operating room, the electrophysiological data, and this is again a study that I'm doing with Adam Zeidel and it's with Israel, the neurosurgeon in Hadassah. And over here we record the electrical activity in the subthalamic nucleus. This is 5 seconds of recording, and this is the tremor of the patient. And you may see very, very clear-cut correlation between the neural activity and the tremor. But what we can do more, we can identify the oscillatory area of the subthalamic nucleus of each patient, for example, by using advanced analysis technique, and to locate our electrode not in the middle of the subthalamic nucleus, but in the middle of the oscillatory area. And what we show over here in a retrospective study, that if we locate the active electrode in the center of the oscillatory area, we get much better effect of the deep brain stimulation. So using this kind of technique, we can already better localize our electrode in the subthalamic nucleus, not in the center of the whole nucleus, but in the center of the oscillatory activity. But even going more far away to the future, we should improve the situation of our patient. And today we put the pulse generator in the chest of the patient, and the patient is coming back to the clinic every second month, every 3 months, and the neurologist adjusts the parameter of the stimulation. But Parkinson's disease is dynamic. The situation of the patient is changing from day to day, from minute to minute. And therefore our dream is actually to test the ability of closed-loop deep brain stimulation system first of all in the monkey, and to record the electrical activity in the brain of the monkey. This is, for example, recording of electrical activity. You may see over here the abnormal activity, but at this point the activity looks very, very normal. So what we can do is to look for whenever there is abnormal activity in the brain, using feedback system and optimization system to adjust the currents that we are injecting to the brain of our patient according to the changes that are occurring every minute. And indeed, with Boris Rovin, MD PhD student, we already start to do it. And we've carried this kind of experiment in 2 monkeys. And over here, looking at the clinical data, this is the amount of movement the monkey is doing when the closed loop circuit is working versus the akinetic before. You may see that with the closed loop we are getting much more movement compared to the standard deep brain stimulation, and of course compared to all other control that we are using that we are not supposed to work, but we like still to work. But the most important is that we get much better clinical improvement compared to the classical, to the standard open loop deep brain stimulation. We all can also look at the electrical activity, and again we are getting much better results over the electrical activity with the closed loop versus the different control that we are doing over here. So our dream for the future is that what we have to do is to indeed to continue and test and approve the superiority of the closed loop DBS in MPTP monkey, then obtain the ethical approval and having the fallback option, because we always can stimulate with the same old method, open loop, if we install closed loop system in the patient, and getting the ethical approval, industrially support, and test closed loop DBS in human Parkinsonian patient. And if closed loop technique will work for us in human Parkinsonian patient, then the sky is the limit. because then we can use different optimization function and probably different target, but going to other basal ganglia and dopamine-related disorder, such treatment-resistant depression, obsessive-compulsive disorder, and maybe even schizophrenia. All these dopamine and basal ganglia disorder need our ability to dynamically change the situation of the patient, and I believe that using such closed-loop technique we'll be able to do it. And the final, this study was carried with many people. One of them is Edmond and that I'm working together for more than 30 years, so I can ensure the Hebrew University that they've done the optimal selection for the new director of the Safra Institute for Brain Research. But just to keep on Bert Sackman saying, I hope along that in the new institute the sign will be that no one that is ignorant of the detail of all the brain, cortex and basal ganglia, and other part of the brain will not be allowed to enter. So thank you.
Thank you very much, Zakai. Let me emphasize one aspect as a mathematician. This is a classic example of multidisciplinary work, because besides, of course, the biological, medical work, the testing, there is very interesting work done into the signal analysis that is involved in analyzing the signals and creating this closed loop. Our last speaker for today will get us even farther into the future, I suspect. You see, all these mechanisms that we are talking now were a try to counteract some deficiency in the activity of the brain. But they were not talking about, maybe too much to expect nowadays, really repairing the brain. One possible approach, we'll see whether it's feasible for repairing the brain, is using stem cells. And one of our top researchers in stem cells technologies is Nissim Benvenisti. Nissim got both his medical degree and PhD, one of the top students in the medical school that do simultaneously MD and PhD at the Wright School at the Hebrew University Hadassah Medical School. Had done postdoctorate in Harvard and came back as a faculty member at the Institute of Life Science. Actually, I had the pleasure of appointing him when I was the Dean of Science. So Nissim will give us a little bit of glimpse of the potential of using stem cells for repairing the brain.
Thank you very much. It's a pleasure to be here, and I will try to tell you today about stem cells and the brain. And in the past 10 years, there were dramatic changes in stem cell research, and Science magazine is choosing every year the breakthrough of the year. Twice in the past decade, embryonic and pluripotent stem cells were chosen as breakthrough of the year, and that will be the topic of my presentation today. So I will briefly tell you what are embryonic stem cells. Then I will go and tell you how we can get nerve cells in culture with the aim, maybe the naive aim, of getting brain in a dish. Then I will go into a little bit of how we can use embryonic development in the creation of structures in the brain, and I will tell you why we needed to use chimera, mixture of two species, something that we try to avoid in research in order to progress our research. And finally, I will try to show you how we can get, maybe generate pattern brain, and I will tell you a little bit about the devastating syndrome, the two-head syndrome, and how we are using that in order to study stem cell research. So when we start thinking about transplantation, we usually think about organ transplantation, but there are many disorders that may benefit just from cell transplantation, and Parkinson is one of them. So basically, if we will be able to isolate the dopaminergic cells and transplant them into Parkinsonian patients, we may be able to solve their problem. It is true also for diabetes mellitus, if we can isolate beta cells and transplant them, for hepatic failure or for problem in the heart muscle. But what are going to be the cells that we are going to use? So basically, stem cells are the building blocks of our body. They are the cells that generate the embryo, and these cells are regenerating the tissues that can be regenerated in our lifetime, like skin or blood. So basically, stem cells have two properties. One of them is proliferation or self-renewal. Stem cells can live forever and proliferate forever. The other capacity is the capacity for differentiation. First, they differentiate into progenitor cells that are still primitive cells, and then into mature cells. The mature cells are the skin cells, the nerve cells, the blood cells. All of them are coming from different stem cells. So in the adult, we still have unipotent stem cells like the stem cells that we have in the male testes that are produced in the sperm. We may have multipotent stem cells like the stem cells in the bone marrow that are creating the white blood cells and the red blood cells. But in my lab, we are studying the pluripotent stem cells, the cells that can generate all the tissues in our body, and those are embryonic stem cells. So we are deriving human embryonic stem cells from very early embryos, from 5-day-old embryos. All of those embryos were created in vitro fertilization clinic. They have never been to the uterus, and all of them were donated by the couples with informed consent under a national health committee. So basically, we are taking early embryos that were produced in a tube, and we are generating embryonic stem cells from them. This is a short movie sequence showing you this early, about 100 cells embryo, and from that we are taking the inner cell mass and growing that in culture until we are getting the culture of embryonic stem cells. This is a colony of embryonic stem cells with about 200 cells. Each circle here is one cell. Those are the tiniest cells in our body and most fast-growing cells, and in my opinion, the most fascinating cells, as you will see in a moment. So basically, can we go and differentiate nerve cells from them? And the idea is that because in the adult, we lack many of the adult stem cells, and even the one that we have, it's very difficult to produce many cells from them, large number of cells from them. We are going to the most primitive cells, the embryonic stem cells, the idea that we will differentiate them in culture and then transplant them into humans. So many years ago, about a decade ago, we have shown for the first time that we can differentiate human embryonic stem cells in culture, in vitro, through the production of embryo bodies. Those are fascinating structures, starting just with a cluster of cells, but after 2 weeks we are getting kind of balloons floating in culture, and in those balloons we have all kinds of cells, blood cells, liver cells, heart muscle cells, as you can see here. This is a human embryo body growing in a tube, and in it we have muscle cells pulsating in rhythm. This is the first documented human embryo body that is pulsating, and we can get nerve cells, which is the focus of my presentation today. And with these neuron cells, we can get different type of neurons, among them also dopaminergic cells. So basically, we could show that with human embryonic stem cells, with simple protocols, by adding different growth factors to the human embryonic stem cells, we can get brain cells, skin cells, blood cells, heart cells, etc. So the basic idea that already was shown in animals is if we differentiate human embryonic stem cells into neurons or to dopaminergic neurons, we can inject them into the brain of Parkinsonian mice, and then they will integrate and improve their symptoms. But our brain is not just a cluster of cells. We are starting as a plate of neurons, but eventually we are creating a tube. And the question was whether we will be able not just to differentiate human embryonic stem cells into nerve cells, can we create a tube from human embryonic stem cells? So to do that, we went to inject human embryonic stem cells, or partially differentiated one, into the chick embryo. So in the chick, we are cutting a piece from the shell, and we are injecting that into the chick embryo. And here you can see the neural tube of the chick, that is the brain and the spinal cord of the chick, and we injected the green cells of human origin next to the neural tube of the chick. When we did it, this is the neural tube of the chick. There is kind of a hole here. And next to that, you can see a very primitive neural tube called neural rosette of a human origin, meaning that the developmental signals that were secreted by the structures next to the neural tube in the chick allowed also the generation of a tube-like structure from human origin. If you look at that, now we can do this kind of tube structures without needing going back to these chimera experiments in the chick. We now know how to do that in culture as well. But if you look at that, this is a tube, but it's very symmetric. In this tube, neural tube of the chick, you can see that there are different structures. The floor plate, the roof plate can regenerate different patterns. And for that, we need to generate an organizer. Many years ago, about 80 years ago, it was shown in the newt that there are a bunch of cells called organizers that are responsible for our anterior posterior axis, meaning that we have only one head, one brain. But when Edmond injected these cells, the organizer cells, in two different places, he got a two-head newt. In humans, we have a very devastating and extremely rare syndrome that has the same phenotype. It's not siamese twins. Siamese twins are fused embryos. Those are embryos with two heads, with two spinal cord, and the idea is that they may have two organizers. So an organizer is allowing a gradient of factors, and by that we know where is the head and where is the tail. But basically, can we do that in our embryoid bodies that are generated from embryonic stem cells? So in our embryoid bodies, again, instead of getting something that's very structured, we are getting many, many tissues, but in a mixture. Is it because we are missing an organizer, or maybe we have too many organizers? By identifying the genes that are involved in the human organizer, we could show that we have organizer cells in our embryoid bodies, but probably we have more than we need, and maybe in different regions. So we've isolated the organizer cells and separated them from the non-organizer cells, and analyzed all the genes in the human genome that are expressed in those human organizer cells, and eventually took these organizer cells back into the chicken frog, and now instead of getting one axis in the chicken, we are getting a second neural tube, meaning that we may have isolated a human organizer that may now enable us not just to get nerve cells or a tube, but maybe more of a pattern tube. So our aim is now forcing the embryo body, instead of making multiple organizers, making only one organizer. So that was just briefly on the topic of our research on how we are trying to get the brain in a dish. And it's clear that in the field of stem cells, we got so far in about 10 years. Now, in many of our labs, in the labs in the world, including in ours, we are isolating and establishing new lines of pluripotent cells, not just from embryos. We can do that now by induced pluripotency from skin cells.